So today's topic is development and manufacturing strategy for MR template plasmids. Um, Here is my top talk's main contents of this talk. So first of all, I'd like to introduce the plasmid applications in gene and cell therapy. So actually, plasmid DNA have very complicated applications in gene and cell therapy. So here we can see there are two main scenarios. So in the first scenario, plasmids is treated as biological drugs. So it's quite easy to understand that plasmids can be used as biopharmaceuticals, for example, as DNA vaccines or other components of in vivo non-viral gene therapy or editing platforms. And in another case, as plasmid treated as starting material. This is a more widely used scenario, and plasmid can be either be used for ex vivo genetic engineering cell, or it can be used to manufacture the viral vectors, mRNA, which eventually turns to drug substance for bio, biopharmaceuticals use. And in these slides, we made a brief summary about plasmid types, features, applications, and several clinical cases. First of all, it is the supercoiled plasmid. It's the most widely used in gene cell therapy. Usually, it shows high yield and um, the its production process usually is scalable and robustness. It serves as raw material for gene and cell therapy or MR therapeutics. For the regarding clinical cases, so we quite will know that the cavity from Legend and J&J is one of the most successful CAR-T therapies. And supercoiled plasmid encodes genes for lentiviral vectors and T-cell engineering. Another gene therapy cases, the Logisma from Novartis, yeah, it's a V vector-based medicine. And because this today's talk is about the mRNA templates, mRNA template plasmids. So linearized DNA actually is known as the starting point templates of the IVT for mRNA. So yeah, it's it's the regarding the clinical cases. So it will know that the COVID-19 mRNA vaccines and the RSV vaccines from Moderna, it's widely used in the commercial stage. And also there are also other types of plasmids, such as the mini circle nanoplasmids, gene circles, which mark-free plasmid is much smaller than traditional plasmid, so it has reduced the transfection toxicity and much safer. And also regarding the linear closed DNA, it only consists of the GOI without bacterial sequences. It's a very good donor plasmid for gene editing and cell therapy. Similarly, the single strand DNA can also work as the donor for gene editing. And the next part, I'm going to talk about the mRNA template plasmids manufacturing challenges and strategy. So this slide, I think, is the whole picture for plasmid manufacturing challenges in different applications. So we list some key challenges here and strategies as well. We can see here there are different features of plasmid. For example, for the mRNA therapeutics and vaccine application. So this mRNA template plasmid has a long poly-A tail, and also for the AV scenario, AV plasmids actually has the ITR structure, and this special structure actually will lead to the challenges um, during the plasmid manufacturing about how to keep this structure integrity during the whole manufacturing. Um, Also, yeah, due to the different scenario, they are different demanding in high yield and low cost. Yeah, uh, as well. Because today's topic is about MR template plasmid, so I'm going to focusing on this the key challenge for the MR template plasmid. So the for this category of plasmid, in this scenario, the key challenges is focused on poly A and high yield. And ProBio has well prepared some solutions to will solve these challenges. For example, we can provide dedicated backbone and proprietary strain together with the specific upstream and downstream process. So in next part, I'm going to give some detailed case study. First of all is the case study about how to keep polyethyl stability. So this slide highlights a critical challenge in our template plasmids manufacturing. So maintaining poly-A integrity and stability uh, is quite important. The polyethyl is a key determinant of mRNA stability and translational efficiency, and also like protein expression. Because IVT mRNA is transcribed directly from plasmid DNA templates, so any truncation or deletion in the plasmid encoded poly-A region is will be directly reflected in the resulting mRNA and leading to the poly-A lens heterogeneity and the poor product quality. The main issue is that long poly-A sequences are highly repetitive and unstable during replication in E. coli, making them tend to shorten or partial deletion. As illustrated in the chart, intact and pure poly-A tail in plasmids directly result in high mRNA poly-A purity. Therefore, maintaining plasmid poly-A integrity is very essential for achieving high quality mRNA manufacturing. And then this slide highlights our strategy to overcome poly-A instability through ProBio's proprietary engineered strain, the Power S PolyAssure. So compared with commercial strain, Power S PolyAssure demonstrates multiple improved positive clone rate across different mRNA templates, plasmids, and achieved plasmid poly-A purity above 95%. The sequencing data further confirm its super ability to maintain poly-A integrity during E. coli passage, ensuring greater sequence stability for long poly-A region. In addition, Lee strain delivers robust fermentation performance. It even can reach yield of up to 5,000 milligram per liter while still sustaining strong cell growth. Overall, PowerS Poly A Assure provides a robust and scalable platform for high quality MR template plasmid manufacturing. Here, yeah, we present our integrated strategy combining ProBio's proprietary plasmid backbone with the Power S strain series to further enhance poly A stability. By optimizing both the vector backbone and the host strand, we achieve significant improvements in positive clone rate, the plasmid poly A purity, and final mRNA poly A purity compared to the commercial strand. The sequencing data also confirm strong poly A stability during E. coli passage, while fermentation performance is sustainably substantially, yeah, improved, with yield reaching up to 1,000 milliliter per liter. Yeah, it's a quite impressive the yield. So together, this combination can um, provides a more robust and scalable solution for high quality mRNA templates plasmid manufacturing. And the next, I'm going to talk another case study about how to increase the fermentation yield. So here we list all the historical data of fermentation yield and uh, the total recovery of the linearized plasmids. So we can see here that for ex vivo mRNA application, the yield of the mRNA template plasmid is higher than the 300 milligram per liter, and for the in vivo mRNA application, so most of them is higher than the 600 milligram per liter, and the total recovery rate of linearized plasmid using our traditional purification step is really is around 15%. So yeah, here we represents presents our upgraded upstream development strategy to improve fermentation yield in mRNA template plasmid manufacturing. So we can see here after process optimization, we achieved significant improvement in fermentation performance across multiple projects. Plasmid yield increased from lower than 600 milligram per liter to over 600 milligram per liter, with the highest reaching to up to 1.7 gram per liter. At the same time, cell growth and the specific plasmid yields were both enhanced, demonstrating improved overall process efficiency. Importantly, poly purity still remained stable throughout the entire manufacturing process, ensure the product quality while increasing the productivity together. Yeah. And the next slides, I'm going to give another case study to how to reduce the cost of the plasmid DNA used for mRNA application. So actually, here we show the two different process 1.0 and ProPlus uh, process. So ProBio has upgraded our this traditional process 1.0 to ProPlus process. The main difference here is between these two processes is that ProPlus is a continuous process which starts from the fermentation and directly ends up with nearest DNA. We don't have to manufacture the circular plasmid DNA first and QC, followed by the linearized process and QC. So therefore, the recovery rate is highly increased and turnaround time is decreased, while still the cost, yeah, heavily reduced. Um, the the traditional linearized DNA process um, um, from mRNA template plasmids contains multiple steps. So yeah, and long production turnaround time, creating a critical bottleneck in the mRNA manufacturing. So this leads to the extended timeline and high cost for research and clinical supply. Our ProPlus process significantly reduce process complexity and production cost while still shortening timeline and increase in yield. So there are some key points here is there are two key challenges, key changes from the ProPlus to the process 1.0. So first of all, we simplify downstream process. So directly captured plasmid DNA from bacterial lysate and then reduce them uh, reduce the need for lysated ultrafiltration and geofiltration chromatography. And secondly, there's no circular DNA QC release. We manage, so in ProPlus, we manage circular plus DNA as an intermediately and only perform in process control testing instead of the full QC panel to release circular plus DNA. And as a result, we can is it yeah, the ProPlus process can increase the yield of linear DNA by 2 to 3 folds compared to the process 1.0. And regarding the timeline, we can shorten the turnaround time from cell banking to final linearized DNA release by as much as 50%. And regarding the cost, yeah, we can achieve like a 20 to 50% reduction of the total cost of goods. Yeah, so this is the big advantages of the ProPlus to really increase the cost and also to decrease the turnaround time. And another case study I want to show here is a very uh, different scenarios that require very fast turnaround time. And let's see how ProBio to solve this problem and these challenges. And yeah, so mRNA vaccine has become a promising platform for cancer immunotherapy due to its high potency, safety, rapid development potentials, and cost-effective manufacturing. So personalized mRNA cancer vaccine, we'll call it PCV. PCV mRNA is one of the mRNA vaccines that provides a new direction for precision tumor treatment with the support of existing clinical data and a large number of ongoing clinical trials. But the key challenges for this PCV mRNA scenario is you can see here, it requires 6 weeks for manufacturing. It's also in this scenario, actually each patient have different sequence. So it means 6 weeks from the sequencing, the mRNA manufacturing and the plasmid manufacturing and mRNA manufacturing is quite challenging. Yeah, so it's 6 weeks it's almost impossible for the E. coli manufacturing to achieve this challenging goal. So in order to solve this issue, ProBio developed cell-free DNA synthesis technology. We call it PreSyn. So PreSyn is for linearized DNA production. Our proprietary PreSyn technology utilizes rolling circle amplification, RCA. There's no cell banking and no fermentation process, which simplifies the whole manufacturing just in a very rapid way. And we can finish this just in 2 or 3 days. So pursuing cell-free technology enables production of linearized DNA with an easier protocol and at a faster speed. Turnaround time of processing process can be as fast as 3 weeks, including gene synthesis, RCA, linearized DNA purification, and release, while it takes at least 5 weeks for E. coli process. And this slide highlights the performance of protein linearized plasmids in key steps of mRNA vaccine production using the PCV as a case study. So the data compared two types of linearized DNA templates, E. coli derived versus cell-free derived across critical quality attributes. So protein linearized plasmid shows comparable quality to E. coli derived linearized plasmids. So regarding the IVT mRNA quality, they are also comparable in this critical quality attributes. For example, integrity and purity as the mRNA label. So this demonstrates the robustness of protein platform for rapid, high-quality mRNA template production, addressing the manufacturing challenge of PCV vaccines. So yeah, finally, just a summary for this talk. So the main challenges to develop and manufacture mR template plasmids are usually focusing on how to maintain the poly integrity from strain selection to the fermentation. Also, how to continuously to increase the fermentation yield to reduce the cost and also to maintain the very fast deliver timeline is the key challenge here. So ProBio will prepare the whole toolbox from the sequence design to the manufacturing to the very specific manufacturing process. And so starting from the backbone, ProBio has our own proprietary backbone, and compared to backbone to starting from the sequence design to avoid the risk of the poly A losses. And also this backbone combined with our proprietary strain can like 100% secure the integrity of poly A tail. And no matter how long the poly tail is and how difficult this sequence is, and finally, the unique process like the ProPlus and ProSyn process to provide competitive the choices to meet varied needs in different application scenarios. Yeah, so that's all for my today's talk. Thank you.